Hello and welcome, dear students. Today we are going to discuss the topic non-Mendelian inheritance and complex diseases. Dear students, the main objectives of today's lecture are to know the basics of inheritance patterns, to understand the non-Mendelian inheritance pattern, to understand the essential features of inheritance by organelles and organelle DNA, to explain the examples of non-Mendelian inheritance, to explain the examples for the mitochondrial and plastid inheritance, to discuss the non-Mendelian inheritance and its relation to complex diseases. Dear students, let's first start with the introduction to non-Mendelian inheritance. Non-Mendelian inheritance is any pattern in which traits do not segregate in accordance with Mendel's laws. Nuclear genes govern most of the characters of an individual. However, some of the traits may be controlled by extranuclear factors or genes located outside the chromosome or nucleus. Non-Mendelian inheritance or cytoplasmic inheritance or extranuclear inheritance or extrachromosomal inheritance or organelle inheritance is the one in which the transmission of characters from parents to offsprings is governed by non-Mendelian genes or extranuclear genes or organelle genes. The best example of non-Mendelian inheritance in animal cells is the mitochondrial genome, and in case of plants, it is plastid, that is the chloroplast genome. The factors governing the non-Mendelian inheritance are called plasmon or plasmogenes, which are present in the chloroplast, that is cpDNA, or mitochondria, that is mtDNA. The characters which are inherited by plasmon genes are inherited in uniparental fashion by the female parent only. Therefore, it shows reciprocal differences in the F1 generation. The presence of DNA in chloroplast in plant cells was first demonstrated in 1962 by Riss and Polt, while as a year later in 1963, the presence of DNA in mitochondria was proved by Margit M.K. Nass and his co-workers. Further observations from time to time by several scientists have been reported about the critical role of cytoplasm in genetics. Although chromosomal genes specify the vast majority of mitochondrial proteins, but the mitochondrial genome has considerable variability and mutates more readily than chromosomal genes, in part due to poor proofreading during DNA synthesis. The resulting mutations are the source of considerable, that is non-Mendelian genetic disease, as well as phenotypic variation in normal mitochondria function. The circular mitochondrial genome with approximately 16,000 base pairs has genes for ribosomal RNA, transfer RNA, and approximately a dozen mitochondrial proteins, including a polymerase. Non-Mendelian inheritance also occurs for centrosomes, usually via the sperm in mammals. Non-Mendelian inheritance of viruses occurs in some situations. In these cases, there is nucleic acid sequence specifying a non-Mendelian component. Dear students, let's discuss the types of non-Mendelian inheritance. First, the incomplete dominance. In this type, the principle of dominance as discovered by Mendel is not applicable. However, the principle of uniformity is seen. In the incomplete dominance, the genetic traits mix, which produces an intermediate phenotype in terms of physical traits. The pink rose is a great example in which the white and red varieties of rose are hybridized and the resulting offspring is pink rose. Dear students, now the codominance. In an offspring of an organism, if we see traits from two alleles, then it is a result of codominance. The blood group in humans is a good example of non-Mendelian genetics codominance. Someone with blood group AB expresses the allele of both blood groups A and B. That means on the RBC membrane, both the antigen A and B are present. Another example of codominant inheritance is in varieties of domestic fowl or chicken. In them, the varieties of black and white feathers are codominant, and when the fowls of both these separate traits are bred, then the offspring show both white and black feathers. Dear students, let's discuss the subtypes of codominance. First, the multiple allelism or multiple alleles. Some population shows the presence of multiple alleles of one gene. For example, in rabbits, there is a C gene that defines the color of coat in the breed. There are 4 common alleles of this gene, like capital C, giving black or brown fur. Then there is capital C, small C, small ch, giving gray fur color, also known as chinchilla coloration. CH, giving white body fur and dark ears, face, feet and tail. And small c, that is recessive, giving a pure white fur and reddish eyes as a result of albinism. Dear students, let's now discuss another example of codominance, that is pleiotropy. In pleiotropy, one gene affects multiple characteristics of the individual. The disease phenylketonuria is an example. It originates from the defect of a single gene on chromosome 12. However, it has an impact on multiple systems like the integumentary system of the skin and nervous system. Albinism is another example of one gene affecting the skin, eyes and hair colors. Let's now discuss another example, that is lethality due to alleles. At times, the combination of the multiple alleles can make the survival of an individual difficult to impossible. For example, a hybrid between 2 heterozygote yellow mice makes them give birth to yellow and brown mice in a ratio of 2:1. So mice that have homozygous alleles die during the gestation period, especially during the embryonic development. These kinds of lethal alleles can be found in dominant or recessive forms, and they can express the individual's traits in homozygous or heterozygous cases. Dear students, let's now discuss the polygenic inheritance. There are some traits that are controlled by more than one gene. Height in human beings, for example, is controlled by more than 400 different genes. Similarly, the pigmentation of the skin or skin color is controlled by at least 4 genes. In fruit flies, the reddish brown pigment in the eyes is a result of at least 3 genes. Dear students, let's now discuss the extranuclear inheritance. Extranuclear inheritance is also known as cytoplasmic inheritance and sometimes is also known as mitochondrial inheritance. In this type of inheritance, some DNA of the mitochondria is passed from one mother to the offspring. Although mostly the extranuclear inheritance from the mitochondria, nevertheless it may occur from the chloroplast too. In cloning, for example, there is a risk of transfer of genes from the mitochondria of the donor cell. There are even some genetic disorders that pass from the mother to the offspring that have their origin in the mitochondrial DNA. In the spermatozoa, for example, there is no cytoplasm, therefore the phenotype of the traits related to extranuclear DNA is derived from the mother only. Dear students, let's now discuss the gender-linked inheritance. In this type of non-Mendelian inheritance, we see particular traits or characters in an individual that are related to gender. Disorders like color blindness and hemophilia are genetic and gender-related disorders. Dear students, let's now discuss the non-Mendelian inheritance by cellular organelles. Extranuclear inheritance is defined as the inheritance of traits of only one of the two parents, generally the female parent that is the mother, to the progeny, where the reciprocal crosses show consistent differences as well as there is a lack of segregation in F2 generation and subsequent generations. The true non-Mendelian inheritance is one that involves mitochondria, that is mitochondrial inheritance seen in both plants and animals, and chloroplastid, that is plastid inheritance seen in plants only. The total of the genes present in the cytoplasm of a cell is known as plasmon. All the genes present in a plastid or the genome of the plastid are known as plastomes. In the same way, all the genes present in a mitochondria are called chondriomes. The genes present in plastid and mitochondria are located in their DNA and are known as cpDNA and mtDNA respectively. These DNAs are collectively termed as organelle DNA. Dear students, let's now discuss the non-Mendelian inheritance by mitochondria. Mitochondria can self-replicate and represent another genetic system in the cell. Of course, the amount of mitochondrial DNA is so small, representing less than 1% of the nuclear DNA in mammalian cells, and it can code for a part of the protein in the mitochondria. The genes present in mitochondria in some cases control the inheritance pattern and are transmitted through the cytoplasm of the eggs, that's the female gamete. The synthesis of the cytochrome found in mitochondria, for example, is known to be present in minute amounts in cytoplasm under the control of nuclear genes. Therefore, it is suggested that both mitochondria and chloroplast seem to have a semi-autonomous existence, and their DNA forms the basis for genetic systems separate from that in the nucleus. The examples of mitochondrial inheritance include inheritance of kappa particles in paramecium, pockiness in neurospora, and peptide in yeast. Dear students, let's now discuss the non-Mendelian inheritance by plastids, that is plastid inheritance. The inheritance pattern of plastid characters due to plasma genes located in plastid is known as plastid inheritance. Plastid inheritance was first case of non-Mendelian inheritance to be discovered independently by Correns and Bohr in 1909. The inheritance pattern of the genes located in plastids is best studied in plant like 4 o'clock plant, which is known as Mirabilis jalapa, Zea mays, and Primula sinensis, and the species of Oenothera lemachiana. Dear students, let's now discuss about non-Mendelian inheritance and its genesis. Non-Mendelian inheritance was first discovered by Carl Correns in 1908 while he was working with Mirabilis jalapa plant. Correns observed that leaf color was dependent only on the genotype of the maternal parent. Based on these data, he determined that the trait was transmitted through a character present in the cytoplasm of the ovule. Later on, Ruth Sager and others identified DNA present in chloroplastus as being responsible for the unusual inheritance pattern observed. Work by Mary and Herschel Michel on the pocky strain of the mold Neurospora crassa ultimately led to the discovery of genetic material in the mitochondria. So the mitochondrial DNA. The question of how the mitochondria and chloroplastus came to have these specific sets of genes is still a matter of experimentation and debate in biology. It is generally believed that these two organelles, that is mitochondria and chloroplastus, arose in the course of evolution as endosymbionts. As per the endosymbiontic theory, mitochondria and chloroplastus were once free living organisms that were each taken up by a eukaryotic cell. Specifically, cells of ancestors of eukaryotes were invaded at different times by prokaryotic cells, one of which was photosynthetic, giving rise to chloroplasts, and other of which was non-photosynthetic, giving rise to mitochondria. Over time, mitochondria and chloroplasts formed a symbiotic relationship with their eukaryotic hosts. These invasions set up mutually beneficial symbiosis, which was a critical event in the origin of the lines that eventually became modern eukaryotes. Although the transfer of a number of genes from these organelles to the nucleus prevents them from living independently, each still possesses genetic material in the form of double-stranded DNA. It is the transmission of this organelle DNA that is responsible for the phenomena of extranuclear inheritance. Both mitochondria and chloroplasts are present in cytoplasm of maternal gametes only. Paternal gametes, that is sperm, for example, do not have cytoplasmic mitochondria. Thus, the phenotype of traits linked to these genes found in either mitochondria or chloroplast are determined exclusively by the maternal parent, that is the female or mother. Dear students, let's now discuss the essential features of non-Mendelian DNA. Both nuclear DNA as well as non-Mendelian are liable to mutation. Any mutation in nuclear DNA leads to alteration or change in nuclear genes, and likewise any mutation in non-Mendelian DNA leads to change in the behavior of plasma genes. Second, non-Mendelian DNA is autonomous and is capable of transcription and translation like nuclear DNA. The code only for the limited number of enzymes and polypeptides as well as chlorophyll and other pigments are synthesized in the plastid. Third, the non-Mendelian inheritance is governed by genes that are found in mitochondria and chloroplasts, and these genes are known as plasma genes or non-Mendelian genes or cytogenes or extranuclear genes. These genes are found in chloroplast DNA, cpDNA, and mitochondrial DNA, that is mtDNA. Fourth, the synthesis of nuclear DNA takes place only during interphase, that's S phase of the cell cycle, while as the synthesis of mitochondrial DNA as well as chloroplast DNA continues throughout the cell cycle. Fifth, usually the nuclear DNA is linear in the eukaryotic system and circular in prokaryotes. But generally, the non-Mendelian DNA is a circular molecule except in ciliates, protozoa, where it is a linear molecule. Sixth, the organelle DNA are directly associated with the inheritance of some phenotypes which are not controlled by the nuclear genes. 7. The genetic material of mitochondria and chloroplast is transmitted almost exclusively through the egg or female gamete. Dear students, let's now discuss the significance of non-Mendelian inheritance. Non-Mendelian inheritance has been beneficial in explaining the role of various non-Mendelian organelles in the transmission of characters in different organisms. Studies of non-Mendelian inheritance have played a key role in the mapping of chloroplast and mitochondrial genome in several species such as yeast, Chlamydomonas, maize, and human beings. Development of non-Mendelian male sterility, that is CMS lines, have been developed in many crops like maize, pearl, millet, and cotton. The availability of CMS lines has helped the production of the hybrid seeds in these crops at a cheaper cost than with hand emasculation and pollination methods, whereas the CMS cytoplasm can be easily transferred to many agronomic bases for their use in the development of superior hybrids. The development of non-Mendelian male sterility, several crop plants like maize, pearl, millet, sorghum, and cotton. The role of mitochondria in the manifestation of heterosis and mutation of chloroplast DNA and mitochondrial DNA leads to the generation of new variants. Dear students, let's now discuss some examples of non-Mendelian inheritance. First, the disease infectious heredity. Infectious heredity is another form of non-Mendelian inheritance. Infectious particles such as viruses may infect host cells and continue to reside in the cytoplasm of these cells. If the presence of these particles results in an altered phenotype, then this phenotype may be subsequently transmitted to the progeny. Because this phenotype is dependent only on the presence of the invader in the host cell cytoplasm, inheritance will be determined only by the infected stats of the maternal parent. This will result in a uniparental transmission of the traits or characters, just as in extranuclear or non-Mendelian inheritance. Killer phenomena exhibited in yeast is one of the most well-studied examples of infectious heredity. Two double-stranded RNA viruses designated L and M are responsible for this phenotype. The L virus codes for the capsid protein of both viruses as well as an RNA polymerase. Thus, the M virus can only infect cells already harboring L virus particles. The M viral RNA encodes a toxin that is secreted from the host cell. It kills susceptible cells growing in close proximity to the host. The M viral RNA also renders the host cell immune to the lethal effects of the toxin. For a cell to be susceptible, it must therefore be either uninfected or harbor only the L virus. The L and M viruses are not capable of exiting their host cell through conventional means. They can only transfer from cell to cell when their host undergoes mating. All progeny of a mating involves a doubly infected yeast cell will also be infected with the L and M viruses. Therefore, the killer phenotype will be passed down to all the progeny. Another example is that in Drosophila, heritable traits that result from infection with foreign particles have also been identified. Wild-type flies normally fully recover after being anesthetized with carbon dioxide. Certain lines of flies have been identified that die off after exposure to the compound. This carbon dioxide sensitivity is passed down from mothers to their progeny. This sensitivity is due to infection with sigma virus, a rhabdovirus only capable of infecting Drosophila. Although this process is usually associated with viruses, recent research has shown that the Wolbachia bacterium is also capable of inserting its genome into that of its host. Dear students, let's discuss another example of genomic imprinting. Genomic imprinting represents also an example of non-Mendelian inheritance. Just as in conventional inheritance, genes for a given trait are passed down to progeny from both parents. However, these genes are epigenetically marked before transmission, altering their levels of expression. These imprints are created before gamete formation and are erased during creation of germline cells. Therefore, a new pattern of imprinting can be made with each generation. Genes are imprinted differentially depending on the parental origin of the chromosome that contains them. In mice, the insulin-like growth factor 2 gene undergoes imprinting. The protein encoded by this gene helps to regulate body size. So mice that possess two functional copies of this gene are larger than those with two mutant copies. The size of mice that are heterozygous at this locus depends on the parent from which the wild type allele came. If the functional allele originated from the mother, the offspring will exhibit dwarfism, whereas parental allele will generate a normal-sized mouse. This is because the maternal IGF2 gene is imprinted. Imprinting results in the inactivation of the IGF2 gene on the chromosome passed down by the mother. Differential methylation of paternal and maternal alleles forms the imprints. This results in differing expression between alleles and the two parents. Sites with significant methylation are associated with low levels of gene expression. Higher gene expression is found at unmethylated sites. In this mode of inheritance, phenotype is determined not only by the specific allele transmitted to the offspring, but also by the sex of the parent that transmitted it. Dear students, another example is the trinucleotide repeat disorders. Trinucleotide repeat disorders also follow a non-Mendelian pattern of inheritance. These diseases are all caused by the expansion of microsatellite tandem repeats consisting of a stretch of 3 nucleotides. Typically in individuals, the number of repeated units is relatively low. With each successive generation, there is a chance that the number of repeats will expand. As this occurs, progeny can progress to premutation and ultimately affect states. Individuals with a number of repeats that falls in the premutation range have a good chance of having affected children. Those who progress to affected states will exhibit symptoms of their particular disease. Prominent trinucleotide repeat disorders include Fragile X syndrome and Huntington's disease. In the case of Fragile X syndrome, it is thought that the symptoms result from the increased methylation and accompanying reduced expression of the Fragile X intellectual disability gene in individuals with a sufficient number of repeats. Dear students, another example is gene conversion. A major form of non-Mendelian inheritance is the gene conversion. It arises during DNA repair via DNA recombination, by which a piece of DNA sequence information is transferred from one DNA helix, which remains unchanged, to another DNA helix whose sequence is altered. This may occur as a mismatch repair between the strands of DNA which are derived from different parents. Thus, the mismatch repair can convert one allele into another one. This phenomena can be detected through the offspring non-Mendelian ratios and is frequently observed in fungal crosses. Dear students, let's now conclude the lecture. Organelle DNA plays an essential role in inheriting certain traits or characters in organisms, and it shows that the variant phenotypes caused by the mutations in organelle DNA are generally inherited maternally, that's by mothers. Dear students, it was all about today's lecture regarding non-Mendelian inheritance. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.